Hello guys, I am Sawan from Inspiring Minds, and today we are going to discuss about the sympathetic pharmacology. See, relatively it is an easy topic, but it's very confusing for the students to understand because they are mostly confused about receptors and what are these receptors doing, the sympathetic receptors. So let us discuss and I'll give you an introduction. What is sympathetic system? It is like the parasympathetic system. The structure is the same. Okay, the spinal cord. Okay, you can imagine spinal cord. Okay, and from the spinal cord, preganglionic fibers are coming. Preganglionic fibers are going in the ganglion, and that ganglion is called as sympathetic ganglion. And from the sympathetic ganglion, postganglionic fibers are coming, and these postganglionic fibers fire on what? Effector organs. And what does this postganglionic neuron secrete? They secrete norepinephrine. So mostly the postganglionic sympathetic fibers, they are secreting norepinephrine on the effector organ. So this norepinephrine is secreted by, this is a neuron, yes, and this norepinephrine will be secreted in the synaptic cleft, and from the synaptic cleft this norepinephrine will bind to the target organ. Wherever the receptor of the norepinephrine will be present, it will bind there. Okay, sympathetic system is what we say, it sympathizes with you. Okay, it get activated when you are in a fight, flight or fright mode, means an emergency mode. For example, a dog is behind you, so and you want to run away, so there should be some changes taking place in your body in order to adapt to that fearful situation. So that changes that takes place in your body, it activates the sympathetic system. So for example, your heartbeat will be very fast, your breathing rate will increase, your pupils will dilate, your skeletal muscles will have more blood supply because you want to run away, and your bladder, that is urinary bladder, it will relax because you don't want to pee, you don't want to urinate if the dog is behind you. So these are the changes that occurs. Blood supply is also distributed. For example, more blood should reach your heart, more blood should reach your brain, and more blood should reach your skeletal muscles, because these are the organs that will help you to get out of this stress. The dog is behind you, and the blood supply to the kidney is reduced, to visceral organs are reduced, for example gastrointestinal tract, because you do not need blood supply there if the dog is behind you. Okay, so that is the situation where sympathetic system is going to be activated. We'll study about the receptors and how is the norepinephrine made, because norepinephrine is a key neurotransmitter in the sympathetic nervous system. Okay, sympathetic activity. So let us understand. This is a neuron, ending of a neuron, or you can say a sympathetic neuron, maybe postganglionic. Yes, it is postganglionic neuron. And what do you need to have? You need to have norepinephrine, because this neuron want to secrete norepinephrine to the effector organs. Okay, so now we are going to study how this neuron, this postganglionic neuron is able to make norepinephrine. So let us study. So this is the membrane of the neuron, this black one, and as you can see, there is a special transporter here that is called a sodium tyrosine cotransporter. So tyrosine, you should remember, tyrosine is the basic product, your raw product that you need for the manufacturing of norepinephrine. So the tyrosine will come inside this neuron. Okay, so this is the tyrosine that has come inside the neuron through this channel, or you can see the transporter. Okay, and the tyrosine is there. This tyrosine is now acted by which enzyme? Tyrosine hydroxylase, this red enzyme, tyrosine hydroxylase. It is the first step. Okay, tyrosine is acted by tyrosine hydroxylase, and the tyrosine is converted into dopa. Tyrosine kya hai? It is a type of amino acid. And what is this enzyme tyrosine hydroxylase doing? Hydroxylase means it is adding a hydroxyl group, OH group. So tyrosine plus OH group that you have added to the tyrosine, this tyrosine now becomes dopa. Dihydro. So dopa means dihydroxy, some acid. It is a type of also amino acid. Okay, and now you have dopa This dopa should be now converted into dopamine. Okay, dopa, I told you the full form of dopa is dihydroxyphenylalanine. Phenylalanine भी एक अमीनो एसिड है, ठीक है. If you look at the structure of the phenylalanine, एक उसके पास one hydroxyl group is already present on this phenylalanine and one you got by this enzyme tyrosine hydroxylase. So that is why it is now having two hydroxyl groups. So dihydroxyphenylalanine. Okay, this dihydroxyphenylalanine or dopa will be converted into dopamine. Which enzyme is there that is very necessary for this reaction to occur? The second step, decarboxylase. You need to remove a molecule of carbon dioxide from dopa to make it dopamine. So you remove the carbon dioxide by which enzyme? Decarboxylase. So dopa decarboxylase enzyme is needed. Now this dopamine is made. The problem with this dopamine is that in this neuron is, a very sensitive area. This neuron has very naughty cats. You can imagine them as cats. Which are those cats? These cats are monoamine oxidase, mouse. So you can just try to remember, because the cats do meow and meow, this sound, and the MAO enzyme is there, and this MAO enzyme that is present all around here in the neuron, this MAO enzyme wants to eat up monoamines. For example, dopamine is a monoamine. Yes, it is having one amino group. So this enzyme, that is MAO, monoamine oxidase, oxidizes this dopamine. And if you oxidize this dopamine, it is going to be destroyed. It is going to lose its function. So that is why if the dopamine is destroyed by the action of the MAO that are present in the neuron, will you be able to make norepinephrine? No, because dopamine is the precursor to norepinephrine. Yes, so this dopamine should be preserved. It should be in some security room where the MAO cannot perform its action. So dopamine is now transported into the synaptic vesicle. Why do you need to transport? Because to prevent the dopamine from the degradation from the action of MAOs that are present in the neuron. So if the dopamine is transported in these synaptic vesicles, now the dopamine is safe. Now the MAOs, that is monoamine oxidase, cannot act on the dopamine, and thus it is prevented from degradation. Okay, dopamine, I have represented it as DM, dopamine. Okay, now the dopamine in the synaptic vesicle, this vesicle, safe zone for the dopamine. So in these vesicles, the dopamine is going to be acted again by a group of enzyme hydroxylase, dopamine hydroxylase. You will again add a hydroxyl group to dopamine, and it will get converted into norepinephrine. So three enzymes we have talked. First enzyme, tyrosine hydroxylase. So first step is hydroxylase. The second enzyme is decarboxylase, converting the dopa to dopamine, and the third step is again hydroxylase enzyme that is converting the dopamine, that is DM, to NE, norepinephrine. Okay, so you should remember it in series, that first hydroxylase, then carboxylase, decarboxylase, and then we have again action of hydroxylase. Okay, now the norepinephrine is present in your vesicles, synaptic vesicles. Now this norepinephrine obviously should be released in the synaptic cleft, then only it will act on the receptor, and then only the sympathetic effect will be produced. So as soon as the action potential will travel across this neuron, yes, the sympathetic situation will arise. Now the dog is behind you. Okay, your sympathetic nervous system become activated, so action potential is traveling across this neuron membrane, and action potential sensitive calcium channel will open out, and this opening of calcium channel will lead to the calcium present in the extracellular space to go inside the neuron, and this influx of the calcium into this neuron will lead to the fusion of this synaptic vesicle to the presynaptic membrane of the neuron. This vesicle, what we have, it will fuse with the membrane, this membrane of the neuron, that is called as presynaptic membrane. Okay, presynaptic membrane, because it's before the synapse. Okay, so calcium entry is very much necessary for the fusion of the synaptic vesicle with this membrane, and this fusion will lead to the release of the norepinephrine into the cleft. Okay, this is how the norepinephrine is going to be synthesized from the tyrosine and will be released in the synaptic cleft. So each step of this pathway, that is the conversion from the tyrosine till the synthesis of norepinephrine, we why we are studying this pathway? Because we have some drugs and some agents that may act on these enzymes, may block the enzymes or may do something with a whole this pathway. So let us understand which reactions are very important in this pathway. First of all, tyrosine hydroxylase, you should remember this enzyme because it's asked in many questions and all. Because why this tyrosine hydroxylase is very important? Because this enzyme is rate limiting enzyme. Do you understand what is rate limiting enzyme? That enzyme, that is very much necessary for the completion of the reaction. Okay, so most important, rate limiting enzyme, because it limits the rate of this reaction. Okay, so tyrosine hydroxylase, you should know it is the rate limiting enzyme, and if any block to this enzyme occur, any agent blocks this enzyme, so obviously the process of synthesis of norepinephrine will not take place. So do you have any such agent that can block this tyrosine hydroxylase? Yes, and you should remember me tyrosine. So the drug that is there that can block this enzyme tyrosine hydroxylase is me tyrosine. So tyrosine itself is in the name of drug, and you can remember this that this drug has the blocking effect on the tyrosine hydroxylase. So what's the use of this drug? You know, this drug was used as an anti-hypertensive drug. Why? Because it prevents the formation of norepinephrine, and if the norepinephrine is not released, it won't act on the heart, it won't act on the vessel, and the blood pressure will significantly be reduced. So this is an antihypertensive drug, me tyrosine, acting on tyrosine hydroxylase enzyme. The other drug that is acting on decarboxylase enzyme, which enzyme? Dopa decarboxylase, which was converting the dopa to dopamine. Okay, so any drug that can act on dopa decarboxylase, yes, we have, that is Carbidopa. This Carbidopa is given with levodopa in the Parkinsonism treatment. Why? It is very important question, and it can be asked in many ways to you. You know, in Parkinsonism, there is what happens in your brain, substantia nigra, is that there are low level of dopamine in certain part of your brains. So what we do, we give the patient levodopa. Okay, and this levodopa can be converted. Okay, levodopa is analog to dopa, it is just similar to the dopa. Okay, and if you give the patient levodopa, so levodopa should reach his brain, and in his brain the levodopa should convert into dopamine and raise the level of dopamine in the brain. So what happens is that along with the levodopa, we give another drug that is called as carbidopa. Why do we give that drug? Because if we are giving dopa, obviously levodopa, to a patient, and this levodopa in the patient's blood, it can be converted into dopamine in the blood of the patient. Levodopa hasn't reached the brain, and it has already converted into dopamine in the blood of the patient. Why? Because there is peripheral dopa decarboxylase enzymes. Some dopa decarboxylase enzymes, they are present in your blood, maybe here, wherever the drug is absorbing. Yeah, and in the peripheral tissues, this levodopa may be converted into dopamine. And this dopamine, because it is polar molecule now, it cannot cross the blood brain barrier, and it won't be able to reach your brain. So administration of carbidopa prevents the peripheral conversion of levodopa to dopamine, and thus it helps preventing the degradation of dopa. Okay, the third drug we have is reserpine, and this reserpine, what does it do? It inhibits the transport of dopamine into these synaptic vesicles, because dopamine should be transported in the synaptic vesicles, because this transport prevents the dopamine from the action of mouse, the naughty cats. Okay, so inhibition, this drug that is reserpine, it blocks this channel, that is transporter, that causes the transport of the dopamine into the synaptic cleft. So reserpine is an important drug that inhibits the transportation. Carbidopa, I have already discussed that it prevents the peripheral conversion of dopa to dopamine in the blood, and thus levodopa can reach your brain effectively, and in the brain the levodopa can be converted into dopamine and produce good therapeutic effects. Okay, the other drug what we have is guanethidine. What does guanethidine do? It prevents the fusion of these vesicles, these synaptic vesicles having norepinephrine to fuse with the presynaptic membrane of the neuron. Thus, it will prevent the release of norepinephrine into the synaptic cleft. So, that's the main drugs. And now, what we have is, so you should remember these drugs and where the pathway is, working, which enzyme or which pathway is this drugs blocking: reserpine, carbidopa, and tyrosine and guanethidine. Okay, now we are going to study about the fate of norepinephrine. Okay, once the norepinephrine is released, this vesicle is fused here, yeah, and the norepinephrine is released in the synaptic cleft. Once the norepinephrine is released, what should the norepinephrine do now? First of all, norepinephrine can bind to what alpha receptors that are present in the effector organ, or it can bind to the beta receptor, or it can bind to both of the receptors that are there, alpha and the beta. Okay, so effector organ P, it will bind to the receptors of norepinephrine that are present on the effector organs. Maybe it can be anything, on heart, on eye, on GIT. These receptors are present, alpha and the beta. We'll discuss it where they are present, but what will happen with the norepinephrine is that it will bind to the receptor. Okay, the other thing that will happen is the norepinephrine first can bind to alpha receptor, can bind to beta receptor. Other thing what can happen is that norepinephrine can be destroyed within the synaptic cleft. By which enzyme? So it is a question. Norepinephrine can be destroyed within the synaptic cleft by which enzyme? You should remember this sword that I have made here. The name of this sword is COMT, catechol O methyl transferase. This enzyme or this sword that is present within the synaptic cleft degrades the norepinephrine. So norepinephrine can also undergo degradation. It can bind to the effector organ. Other thing that can happen with the norepinephrine is that it can be reuptaken into the neuron, this presynaptic neuron. So norepinephrine, some of it can be reuptaken into the neuron, and for reuptake what do you need? A transporter. Okay, which transporter is necessary? This. What's the name of this transporter through which the norepinephrine will go inside, or it can be reuptaken? The name of this transporter, you should remember, is NET, that is called as norepinephrine transporter, or sometimes it is also called as NAT, that is noradrenaline transporter. Norepinephrine and noradrenaline, it's one and the same thing. So this transporter is very necessary for reuptake of norepinephrine into the presynaptic neuron. Thus, by transporting the norepinephrine back into the neuron, what are you achieving? You are achieving the low level of norepinephrine within the cleft. Okay, so the prolonged action of norepinephrine within the cleft can be prevented by the reuptake of norepinephrine into the neuron, this presynaptic neuron. This the NET transporter or the norepinephrine transporter which I have discussed, it's very important. Why this NET transporter is important? Because you have certain agents that are called as NET blockers. So cocaine is one of the most famous of them. Cocaine, if you are some good guy and if you are in the habit of sniffing the cocaine, so what does this cocaine do? Cocaine inhibits this transporter. So what will happen? The norepinephrine will not go back into the neuron and it will stay in the cleft. So in your CNS, in your central nervous system, the blocking of this transporter will increase the level of what? Norepinephrine within your brain. And this norepinephrine, when it is increased in your brain, it causes what? What does it causes? It causes a tension. The attention of the person increases, the focus of the person increases. He feels so active because of the excessive norepinephrine within the synaptic cleft in the central nervous system. So this is what the effect of cocaine is there. But excessive of it, the excessive effect of the cocaine, the person can become combative, aggressive, and the violent. Okay. And the other drug is the tricyclic antidepressant. Tricyclic antidepressant also increases the level of norepinephrine within your synaptic cleft in the central nervous system, and the high level of the norepinephrine in your central nervous system is antidepressive. This is the basic of the treatment of antidepressive treatment, because अगर हमारा नॉरेपिनेफ्रिन ब्रेन में ज़्यादा होगा, so the excessive of the norepinephrine in the brain is related with the good mood. You won't feel depressed. Same is with the cocaine. Why are you taking the cocaine? Because you are not feeling depressed after that. You are enjoying it. So, okay, but you should not do cocaine because it is addictive. Okay. The other thing we'll discuss about is the receptors, because many students get confused which receptor is this norepinephrine acting on, and it is not very hard to relate the receptors. Okay, just you have to memorize and the effect, what effect is there, and accordingly you have to remember the receptor. For example, first of all, what I like you to memorize is that the alpha 1 and the beta 1, these receptors are stimulatory. Okay, and the alpha 2 and the beta 2, these receptors are inhibitory type of receptors. What do you mean by stimulatory and inhibitory? Alpha 1 stimulatory means that when the norepinephrine is going to bind with the alpha 1 receptor, for example, alpha 1 receptor present on the blood vessels, so it will give signals, okay, to the cells of the blood vessels, smooth muscle of the blood vessel, and it will cause the constriction of the blood vessel. So stimulation of a receptor, the adrenergic receptor, usually means it is constricting some vessel, or it is causing constriction of some smooth muscles. Whereas the inhibitory receptor, for example, beta 2 receptor and the alpha 2 receptors, these are inhibitory receptor. So what they can produce is the relaxation of the vessels, blood vessels, and relaxation of the smooth muscles. So this is the effect you should remember. Stimulatory means constriction of the vessel and constriction of the smooth muscle. Inhibitory means relaxation of the vessel and dilation of the vessel, you can say, and the relaxation of the smooth muscles. So let us know where is this alpha 1 receptor present and why it is present. For example, alpha 1 receptor are present in the blood vessels in your GIT, gut, and the peripheral organs what we have. Why should the alpha 1 receptor is present in the GIT? Because when you are in the sympathetic drive, when you are in the case of flight or flight, you do not want the blood supply to your GIT, you don't want to digest the food. So what do you want to do? You want to limit the blood going there. Okay, so what you'll do, you'll constrict the vessels so that no blood or very less blood is going in your GIT, and that blood is diverted to your brain or skeletal muscles so that you can run away from that dog that is behind you. So alpha 1 is a stimulatory receptor present in the blood vessels, mainly in your GIT, and alpha 1 receptor is also present in your dilator pupillae muscles, the radial muscle of the pupil, because you want to see far away. When you are in the case of emergency, you want to see as far as you can see. So alpha 1 receptor is present in your eye, the pupil, dilator pupillae muscle, and it will cause the dilation of the pupil or the mydriasis. Okay, so it is stimulatory receptor, is causing the contraction of your dilator pupillae muscles. Okay, so again, contraction, stimulation, so it is causing the mydriasis. Okay, and alpha 1 is also present in your internal urethral sphincter. You do not want to pass urine, so the sphincter should be closed or constricted. So alpha 1 is present in your internal urethral sphincter so that you do not pee when the dog is behind you. Okay, so alpha 1 we have discussed. Alpha 2 is present on presynaptic neuron. Okay, And if the norepinephrine from the cleft is binding to the alpha 2 receptor that is present on the presynaptic neuron, what does this cause? This binding of norepinephrine to alpha 2 receptor causes the inhibitory signals to this neuron, and these inhibitory signals will prevent the release of norepinephrine into the synaptic cleft. It is just like a feedback mechanism system. Okay, if the norepinephrine is more in the synaptic cleft, some of the norepinephrine will bind to the alpha 2 receptors on this neuron, presynaptic neuron, and this will cause the less release of norepinephrine now in the synaptic cleft. Okay, and also alpha 2 is present in your pancreas, and it inhibits the insulin release. Why does it inhibit the insulin release? Because when you are sympathetically active in the state of flight or flight, what do you want? You want high glucose level in your blood. So to produce high glucose level in your blood, hyperglycemia in the blood, the insulin is not produced that much, so alpha 2 receptor is present in your pancreas. Then we have the beta 1. Beta 1 is again a stimulatory type of adrenergic receptor. Where it is present, mainly you should remember heart. Okay, so stimulatory, what I told you, what does the stimulatory do? Contract, constrict, okay, pump. So action of norepinephrine on heart will cause the pumping of the heartbeat will increase now. Okay, then it is present on the juxtaglomerular apparatus in the kidney, and the very special type of beta 1 receptor is present on the adipocyte, and this modified type of beta 1 receptor that is present on adipocyte is called as beta 3. Why it is present? Because you need energy, you need glucose, so lipolysis will occur. Degradation of the fat should take place to form glucose, because you need glucose in the state of emergency. When the dog is behind you, you want to run away, you need energy, you need glucose. Okay, then we have beta 2 receptor. Beta 2 receptor is what? Stimulatory or inhibitory? Inhibitory. 2 are inhibitory, alpha 2, beta 2 inhibitory. Okay, so beta 2 का मतलब क्या है? Inhibitory. Inhibitory receptor means relaxation of smooth muscle or the vessels. So the beta 2 receptor is present on mainly the smooth muscles. Which smooth muscles? Your lungs. Okay, the norepinephrine will bind to the beta 2 receptor in your lungs, yeah, the smooth muscle of your lungs, and the bronchus will dilate. Why will it dilate? Because you need to breathe deeply when you are in the state of emergency. Okay, then it is also present on GIT. You do not want the constriction of your GI tract, or you do not want the peristalsis to take place, or you do not want to digest the food when the dog is behind you. So what type of receptor is present in your GIT? Beta 2 receptor. What does it cause? It causes the relaxation of your GIT smooth muscles, so that you do not do the peristalsis. And beta 2, you should remember, it is also present in the some blood vessels. Why it is present in the blood vessels? Because relaxation of those blood vessels will cause what? More supply to the organ. For example, beta 2 receptor is present in your blood vessels that are supplying your skeletal muscles. Dog is behind you, you need high activity of your skeletal muscles to function very well and fast. So what does they need? They need blood for energy, your skeletal muscles, yeah. So how will you provide the skeletal muscles with blood? You will dilate the vessels, and this will cause good blood supply to your skeletal muscles. So beta 2 is present in your skeletal muscles, and beta 2 is also present in your detrusor of your bladder. Detrusor muscle of bladder means जो आपका urinary bladder है, it is present there, and the urinary bladder, it becomes widen, it become relaxed, so that it can accommodate more volume of urine, and it can prevent the release of urine, or to prevent micturition. Okay, so these things you should remember about the sympathetic, how it is formed, which receptors are present where, and why they are present. You again remember everything. Okay, the release, the tyrosine as a product, and then how tyrosine is forming norepinephrine. Then the most important enzyme here is tyrosine hydroxylase, MAO, action of MAO, action of COMT, action of NET transporters. Okay, then NET blockers, you should remember, cocaine and the tricyclic antidepressant. Then the receptors and where they are present. Alpha 1 is present in the blood vessels, okay, and alpha 2 is present in the blood vessel of GIT. Alpha 2 is present presynaptically. Beta 1 is present on the heart. Beta 1 is present on juxtaglomerular apparatus of the kidney. Okay, beta 1 is present on adipocytes. Beta 2 is present on smooth muscles of your lungs. Okay, so thank you for listening to the lecture. Hope you're able to do the questions now. Thank you everybody. Please subscribe and like the videos for future updates.